Hello, good morning. I'll share my.
Good morning. So he comes from a company that deal with oncology and inflammatory disease, and they have a translational view from the research to the patients. And so please, Yinfei, can present your talk that will speak about the shifting of the paradigm of cancer therapy. How can optical imaging help?
Okay, thank you, Claudia, and thank you very much for the organizer gave the opportunity for us to show some of the work we have done. Okay. So basically, over the recent decades, we have seen a paradigm shift in terms of cancer treatment. So chemotherapy and surgical recessions used to be historically the first line of defense, which has later moved more towards targeted therapy. And these approaches all aimed specifically to target tumor or tumor cells. While in recent years, there has been an increased drive in the design of molecules or biologicals that help the immune system to combat cancer. So when used as monotherapies, a great deal has been achieved with immune therapy in terms of patient response and tumor regression, especially with immune checkpoint inhibitors such as PD-1 and PD-L1. So in parallel with advance, there comes limitations such as side effect and resistance. Therefore, recently, there's quite a lot of researchers are focusing on combination therapies. So to achieve a good immune therapy, so we need good models to check them in vitro and in vivo to help us to understand the factors that affect the response to immune therapy and help us. Sorry, I was.
Roy, Villaggi, you're Roy.
Elroy, please close your presentation because this is not your time. We have another presentation. Please don't share your screen.
Okay, so let's carry on. So for immunotherapy models, they can be roughly divided into subcategories. They include models with functional mural immunity. They can be used for the investigation of surrogate and cross-reactive agents, and then the humanized models, which can be used for investigation of human-specific targets. So optical imaging. So using optical imaging, the scientists within Crown have developed quite a few clinical relevant models, and they're mainly including autotopic and metastatic models. So this type of models allows us to better understand the mechanisms of disease, help us to investigate the contribution of clinically relevant organ-specific tumor microenvironment and to treatment efficacy, and also allow us to better understand something like vasculature, stromal interaction, and environmental pressure on tumor. Then metastatic models, they are used to evaluate disease progression, and there's also late-stage metastatic models that can help us to target metastatic lesion, which is probably still most patients died because of this. And then there's a high unmet need to understand them better. So here is a list of models we have developed over the past decade or so. So they cover majority of the cancer types. In the next 15 minutes also, I'm going to use some case studies to go through some data and to showcase what we have been doing. So first, for the models with mural immunity, as mentioned earlier, they are very good to use to test surrogate therapies or cross-reactive agents to help us investigate the MOA of the test compounds. These models, including the CinDNAC models, which are allografts, immortalized mouse cancer cell line, and then the GEM models, which are genetically engineered mouse models with spontaneous tumor. They both have their advantages and disadvantages. I'm going to cover that a bit later. Actually, the team in Crown was trying to find a happy medium. So that's what we developed, what we call the mu-prime tumor homograft. So they are CinDNAC allografts of spontaneous mouse tumors, which has been derived from GEMs. They have the character of GEM and the simplicity operation of CinDNAC. So I'm going to cover that a bit later. So this presentation is all about how do we set up models as isotopic, because we believe they are more clinically relevant. So for CinDNAC, it's the same story. So what we have done is we have labeled quite a few CinDNAC cell lines and with bioluminescence, so we can image them and we can put them where they came from. So there's a large panel of models available at this point, which we have set up. I'm just going to quickly show you some data using the HEPA-16 HCC model. So as we all know, and HCC is a very aggressive cancer with very limited therapeutic treatment options. So immunotherapy, mainly the checkpoint inhibitors, have been evaluated in HCC, but the results are mixed. So we need suitable preclinical models. So orthotopic models, as mentioned briefly earlier, they are set up as organ-specific sites. They provide the tumor microenvironment that's more comparable to patients and better support immune and stromal interaction, which are very important for immune therapy. So using BLI, we can carry out non-invasive longitudinal monitoring of the orthotopic tumor burden. So here's some data. So basically the HEPA-16 murine cell line was labeled with bioluminescence, and then tumor was inoculated into the upper left lobe of the mouse liver. We can monitor the tumor growth with BLI. So that's a vehicle group, that's sorafenib, and that one is anti-PD-1. Both sorafenib, which is the yellow line here, and anti-PD-1 is a purple line here, showed reduced tumor growth. And of course, we can actually take the tumor out, run some flow analysis to check immune cell infiltration. This can also be done with IHC. So once the tumor are taken out of the mice, we can carry out staining and then check the T cell infiltration. So for example, this one is a dual staining or CD4 and CD8 positive. And then we can check in detail, carry out quantitative analysis of this infiltration intensity and how far the immune cell goes. The next two models are quite interesting. So in general, this H22 is another HCC models which I have set up. So we see some really interesting data. As sub-Q, H22 responds really well to sorafenib and anti-CTLA-4. But as isotopic, it doesn't really have any impact. Move to another one is a panel 2 model. It tells us a different story. So at subcutaneous, panel 2 models does response to anti-CTLA-4, while at isotope doesn't, and while at orthotopic, it does. So the data is different between the two setups. So that got us thinking, well, why? What's the difference? So what we have done is we carry out some quantitative proteinomic analysis of the panel 2 tumor. So the analysis revealed about 400 significantly regulated proteins. They are clearly clustered according to the implantation site. So when the unique proteins are identified from each implantation site were analyzed, and they were obviously different in the KAG pathways that were involved. So orthotopic implantation appeared to trigger pathways that were more relevant to the tumor type. So basically pancreatic excretion, metabolic pathways, et cetera, which indicates that models are more clinically relevant. While on the other side, subcutaneously, which most of us actually use, they appear to trigger pathways only associated to more general immune response, which means it's a foreign tumor in the mouse body. That's very much expected. Okay, so that's a quick introduction about the traditional, what we call the synthetic cell lines. Then move to mu prime, we briefly touched earlier. So we all know gems are highly useful for us to study the mechanisms of disease. But the spontaneous nature of the tumors makes them less suitable for studying efficacy of oncology and immuno-oncology agents. So the mu prime is something which we would like to call the happy medium between the synthetic models and gem models. So basically, they are tumors taken from gem mice and preserved in mice. So they still maintain the same genetic driver as gem, but operationally, they are extremely easy to use. They're similar to cell lines. They're fully characterized, and we have genomic pharmacology and molecular data. So to compare, so synthetic models, most of us using them because they are rapid, high throughput, and also sometimes they develop MAT. But mu prime has all that advantage, and with added advantage like they have relevant tumor stroma, they mimic original murine tumor histopathology, and they have disease-specific mutation, which is very important if we want to start a combination therapy. Next few slides, I'm going to quickly show some data using this KPC model. So KPC model carries oncogenic KRAS activation, and it's also TP53 loss. It recapitulates human PDAC tumors. So morphology is small ductal tumor with enriched stromal contents. It has PDAC immune microenvironment. It lacks a response to a series chemotherapies and immune checkpoint inhibitors. So morphology, we can see, so the top left is the patient PDAC, and bottom left is a primary PDAC from KPC, gem mice. The right is subcutaneous and orthotopic mu prime. We can see the structure are all small ductal tumors with enriched stromal contents. So structurally, it's clinically relevant. Then it has also a robust inflammatory microenvironment, has high B cells and macrophages, but very low effector T cells. So when we tried some SOC with gemcitabine and anti-PD-1, PD-L1, so that's gemcitabine, sub-Q orthotopic, and anti-PD-L1, they all have no impact on tumor growth. While using some novel IO treatment, they do see improved survival. The next stage we did is we actually labeled the tumors with luciferase and put them orthotopic as well, so we can monitor the tumor growth longitudinally. So this is some quick data to see. If you look at the very left vertical, that's vehicle, we can see a very nice growth trend. Then when we again treat with, if we concentrate on the graph on the right, we can treat with gemcitabine. We can see this time it responds to gemcitabine, but it still doesn't respond to anti-PD-1. So probably because we have selected a subpopulation of the original tumor. Okay, that's the mouse models, which are murine immune system. Then we move to human tumor models. I'm going to use breast cancer as an example to quickly go through some data. So breast cancer is probably one of the cancers where most progress has been made in the past decade also. So a lot of patients have a very good survival rate, but the metastatic breast cancer is still incurable. And to find new therapies, so we need models to actually model different stages of disease. So breast cancer can be roughly divided into 3 key stages. So primary tumor, then regional lymph nodes, invasion, and distant mat. So the team has actually set up models that can mimic every stage of the development. So we have orthotopic and implantation in the mammary fat pad that will develop spontaneous invasion and then spontaneous mat. But we also have what we call experimental mat that can be used to learn specifically like a brain lesion. So first, the orthotopic inoculation. What we have used is a cell line. Everyone knows it's triple negative, a breast cancer cell line, MDA MB231. It's inoculated into the mammary fat pad, so we can monitor tumor growth using BLI. And we see very good tumor growth. The advantage of this model, it have high incidence of MET. So if we concentrate on a pie chart, we can see there's high occurrence of MET in lung, lymph nodes, slightly lower in liver, and then lower in brain and bone. So this got us thinking a little bit more. And so the model is quite good if you want to study about the lung and the lymph node MET, got a very high incidence. But if you want to focus on brain and bone, the model is probably not suitable because there's high incidence. If you want a large cohort, it's not very practical. So what we have done is we have actually inoculated the cell line intracardiac. So the bloodstream will carry cells around the body. They will settle at bone and develop bone lesion. And again, this can be monitored with BLI imaging. So here's some very quick graph. So on the left, we can see there's multiple lesions all across the body. Endpoint is probably at about 3 to 4 weeks, and this is mainly driven by the long bone lesion. That's a vehicle. On the right is a treated group. It's representative of the treated group. The mice here is treated with Taxotere. So we can see on the long bone, all the lesions are gone, but there's still bone in central nervous system. This is mainly because Taxotere can't really cross blood-brain barrier. So the last couple of minutes, I'm going to quickly touch on the organoid. This is a very hot topic. The organoids are mini organs, which can be maintained in vitro that recapitulates genomic, morphology, and physiological of tumor. They derived from adult stem cells, and they have multiple cell lineage. And also they're genomically very, very stable. So they're basically very easy to use compared with PDX. Well, PDX is very good, but it takes a long time and cost is very, very high. The key advantage of this is this type of organoids are clinically predictive, and they can be scaled up very easily. They can theoretically manipulate it the same as a cell line. The efficiency is very, very high, and also they can be put back into the in vivo and once the selection stage. So it's a great screening tool for drug screening. So what actually the imaging team is opens a new door for us. It allows us to label the patient-derived tumor. So in the past, if we want to set up a PDX orthotopically, it has always been a challenging experience. We have to terminate mice at different stages to look at the tumor size, or we have to actually just look at the survival. So the data is not very neat and also are limited. But using organoid, as we can play the PDX as we do with the cell line, we can label them quite easily. then that opens a new door for us to actually put a lot of PDX orthotopically. As we discussed, when models set up orthotopically, they tend to be more clinically relevant. So this is a simple proof concept. So we label the PDX with GFP. We can see, we can actually select a single colony and do single cell selection. And then we move to luciferase, which is quite good for in vivo. So organoid engineered was expressed luciferase in this one. So we can see once labeled, the model maintains its character, and so we can move back into the orthotopic setting. Just quickly summary, and using BLI imaging is allow us to basically follow the disease progression and the impact of treatment. It has real-time measurement of in-life tumor establishment and growth in a longitudinal, non-invasive manner. It can be very large scale, it can be very high throughput. Also, it has very high signal to noise ratio that simplifies quantification. All in all, it reduces the number of animals required for the study, and the models are more clinically relevant. And in the long run, it saves time and funding to achieve with the end goal, which is help the patient. Thank you very much. That's all my presentation.
Thank you very much. A very interesting talk. Of course, you present a different model that really can help, and you also compare which is could give the best advance, and to have a model in which the proof of concept of therapy, for example, could be studied. This is really very important.
Yeah.
There are some questions for Yinfei. No, we don't have a written question. We because of the short time, we can anyway, as I told, we can interact with each other by chat, by mail, and so we will go through other aspects who is interested also in these models. So we thank you, Yinfei.
Thank you very much.
Thank you.